I will start making you a question. So what comes to your mind when we talk about bioinformatics? Probably it's kind of black box or something like a Manhattan plot if you work with the SNPs, gene expression volcano plot, PCA. This is the kind of things that comes to our mind, no? Gene set enrichment analysis. If we work on other technologies like ChIP-seq or ATAC-seq, then we have the peaks, the profile, what it's opening, closing, interaction between the chromatin, then correlation. Also, now in single cell, how we annotate the cells, we can move to trajectory analysis, and also the networks that all of us love, but we really don't understand yet. So, my next question is, do you know when all this revolution of bioinformatics, that it's around all the biomedical projects nowadays start? How we arrived to that, to the bioinformatic file? So let me show the Big Bang of bioinformatics, okay? So let's say bioinformatics, that's the world say, it's something about the history of biology, but also about the history of bioinformatics. So let's travel in time. This is biology, and probably most of you know. So from the beginning, the laws of genetics, then start the structure of the DNA, the kind of revolution of sequencing. But to reach this synergy between biology and informatics, we need to know also a little bit of the history of informatics. That it starts, this is quite nice, no? We have the first computer in 1946 in the US, thanks to a lot of money for army, etc. But it was not until 1972 that the first microcomputers appear, and it becomes commercialized, and then it arrives to the desktop computers. Then the next big point that was not so far away, it's the World Wide Web. And all this reached to nowadays. So any of you wants to make a bet about when this synergy appears? When do you think that all this kind of revolution starts? Any idea? So what will you, I am going to give you 2 hints. These are Frederick Sangers, that will be key on that, and also Per Victor Edman. I don't know if you know about these guys, but what happened with these guys is that around 1950, they were in a race regarding the protein sequencing. So on 1950, Edman developed the Edman degradation method that allow us to know the sequence of a protein, of the different amino acids of a protein. And over that time, Sanger, that get the Nobel Prize for that, he fully sequenced the bovine insulin. And that was a kind of revolution. But what was more important was that the method of Edman can be automatized. And with this, they start to sequence a lot of protein. But as you can imagine, technology is not perfect. So they were not able to fully sequence the sequence of one protein. They can allow, so they were able more or less to sequence about 30, 50 amino acids, but not more. So, and this became the first challenge, that they think, okay, let's split with different enzymes the protein, and then try to see the overlaps, the cutting with the different enzymes, and then we can get the fully sequence of that protein. So that was the first problem. They got the sequences, but then they need to put in order, identify how they overlap to find the final, the complete protein sequence. And we can do this manually. But then it was in the starting of the computer revolution, etc. And there's a team of people, there's this synergy that developed the first algorithm that is called COMPROTEIN, that allow the determination of the primary structure of the protein based on these fragments. So you can see here, it looks like quite simple, but what this code does with this alienation of the different peptides that the machine gives. And behind that, there's so much of algorithmic definitions. So all of this is from the original paper. It's something simple, but at the end, there's a lot of things behind. So the people that do that was Margaret Oakley-Dayhoff and Robert Stephen Leiden. So now you know, or we know, let's say, that more or less the bioinformatics start on 1962 with the definition of the first algorithm that it's called COMPROT, based on one of, let's say, a need from the biological, medical side to define the structure and know the structure of the proteins. But let me talk a little bit more of Margaret. She was a mathematician, did the PhD on quantum chemistry, started to work a lot of algorithmic programming in Fortran. But this woman wants or has some kind of motivation to translate all her knowledge to the biomedical file. And this is how she arrives to the National Biomedical Research Foundation. And together with Robert, defined the COMPROT. But she not only do that, she also defined the one code letter, the codification of the amino acids that allow these fast and computational approaches for the sequencing, and also develop, together always with other people, but the first atlas of protein sequencing and structure. So it's kind of pioneer on get and put in order all the biological data regarding protein structure, and now we have a lot of databases to ask several questions. But those was a kind of revolution. And for all these achievements, she is considered the mother and the father of the bioinformatics. And at that point, we start to get a lot of sequences of proteins. And what happened with this step forward, it's that these two guys, Emily and Linus, think, okay, we can start to study the similarity between the structure of the proteins. And then they move on in the novel branch of the evolutionary biology, but it's the paleogenomics, and they start to compare that. But they are not going to do by hand. So behind, there's another guy, the Mr. Needleman, that developed the pairwise protein sequence alignment that allow this comparison between the sequences and make which is the best alignment when we compare. So they are going hand by hand, the biology and the bioinformatics, let's say, to move forward. And if we go further, this evolved to a more complex question. It's just, we are not going to compare just two sequences. We want to compare multiple sequences. And here it's where Russell Dolittle developed the multi-sequence alignment. And this, it's the basis for the sequencing alignment that still used nowadays, that it's called CLASTOL. Not our CLASTOL, okay? This CLASTOL, okay? If you go to the ABI, you can use it. So it's something that was well-defined, and it's still ongoing. So just to recapitulate, we can have the sequence of the proteins, the protein atlas. We can start to compare the sequences, and this is kind of revolution. But okay, all of this, it's about proteins. Now we know that there's a relation between proteins, RNA, DNA, but it was defined in 1950 by Francis Crick with the central dogma that define this relation between the different omics that we say today, no? DNA, RNA, protein. But at that time, there was just a sequence of nucleotides. They don't know what it's that. And it was later, with the effort of these mainly three guys, that they deciphered the genetic code and start to have sense of these sequences of DNA. They can translate to proteins. But until that point, we were not able to understand the genetic code. So for that, in 1968, as we have the key to understand the genome, if you see in the plot, the sequencing revolution. And this gives us to the nowadays, the sequence that we are sequencing all we can, different omics, etc. But at the end of the day, what we are having from the beginning, it's biological data. That, okay, we have a lot of data, but what we aim for is to get biological information, not just data. If we are not able to understand the data, it means nothing. And here it's where bioinformatics help us. All these methods help us to get information from the data. So comproteins looks like something like a child's play nowadays, but the challenge that we have, it's much more difficult. We have technologically advanced a lot, and we are able to get information from a single cell, not also the single cell information, also the spatial location of these cells, also how they interact with other cells, and in different layers of cellular information. So our challenge to move from biological data to biological information is much more difficult. And one can think, okay, we have the AI. That's easy. Now, super smart, okay. If I talk about expression heat map, I think all of us have the same image. I asked for expression heat map plot to the AI, and this was, he showed me. So I love it, but it's totally crazy. So we need to take care how we use the bioinformatics, the AI, because as our questions are more complex, the way to interpret, to identify the message that it's behind, it's more and more challenging. And this is why I think this picture is perfect. So our consortium, it's kind of a mirror of what we need. So to go for get information from the biological data that we are generating, we need the clinicians inside to know really the patients, the disease, all the things, how we collect the data, the person that do the data management, how we have the data properly collected and harmonized to then make the correct questions, all the data quality, preprocessing and analysis, because each technique is different, and we need to know which technique we can use regarding the question that we have. Not all the things are allowed. So there's limitation, each technology has the limitations, but all of us need to know the limitation that we have at each point. And finally, we need the synergy of all our expertise to really interpret the data. So yeah, this is our traveling time. Now, Christina is going to show you a little bit the data management challenges.
I will start with a kind of metaphor about what is harmonization. So, is there any musician here in the room? At least one. So, at least one. So, Giacomo, who is that guy? Yeah, and who is that guy? The father of the music, no? Yeah, and because he did the fugues, and the fugues are like, well, if we read, music is a compositional procedure characterized by the systematic imitation of a principal theme and the melodic lines, the counterpoints. So I have done a metaphor saying that, okay, imagine that our canonic, our predict, and our acclara are canonic, let's say, our main theme, and then predict and acclara the counterpoints. So if you see, apparently, you see the colors on the score, they are like the same, because if you read, it's like the same, but at the end, they are not the same, and they are so difficult to harmonize and to meet to put together as a whole. So, what is integration? The action or process of combining two or more things in an effective way. This is also important, because the effective way, sometimes when we start to work in decision with Navarra, Biomed is like the effective way is not what we expected from each other, no? So, and it represented a lot of challenges. So, for example, I will go in deep in all these challenges, putting specific examples of what we have found in the Canonic Predict and Aclara integration. So, white or vertical? Maybe for you is like, okay, I don't care if I work in white or vertical, but when you have to manage several variables, I can assure that vertical is too easy in comparison. Which visits and what about the visit window? So if you have work on Canonic Predict and Aclara, think about the visit windows and what we have as a common visits to harmonize. Every single variable of each cohort, do you integrate? You will integrate this or only, I don't know, several variables. Different categories for same variables among the different cohorts. This is also a challenge because sometimes you have the word, I don't know, ascites, and you have different categories among the, let's say the evolution of the CRFs at the end provokes different categories. What if there is a mistake in the original cohort? What would you do? Because Canonic, for example, or even Predict, they are like from 6-7 years ago, and Canonic from 2011. So difficult also. We have updated some variables. It's like we maintain the first one, we maintain more variables, we update it, we mix, like what we should do. Or should these databases, new databases, be prepared for adding, the integrated database be prepared for adding new databases? This is also an important question when we started to work on this process of integration. And I have to say that after 4 years, since the session started, we are continuing checking almost every week and updating the integrated database because we have found a lot of issues, not issues only, but we have to add and to modify a lot of things. So, white versus vertical. So the image we have up is the integrated database. As you see, we have the patient ID, the visit, the medication, the category, the indication, and the name. With these 6 variables, we have all what we need to analyze the medication data. And the image below is an image from the Predict database. So more or less, we have here like 13 variables. But all these 13 variables are talking only about beta blockers and at hospital admission. Imagine if I have sent to David and Nuria and Sarah, I don't know, maybe 500 variables already for treatments. It's fun, eh? So, the visits, the Canonic design we have for all the groups, because this is also important. It represents that Canonic was the definition of the syndrome Predict, when we identified the precipitating events of ACLF, and Aclara was like a kind of replica to see the syndrome in the Latin American cohort, no? But every single study, when you say group 1 or group 2 or group 3, means a different thing. For example, in Predict, when you talk about group 1, we're talking the ones who had an AD less than 50. In Aclara, when you talk about group 1, means that they are the patients that does not develop ACLF during the whole study. So this is something that should be also harmonized. And also, for example, the group, the visit window, what I was saying before, we have day 3 to 7 in Canonic, for example, and day 8 to 14. But now we are in Predict, and we have the week 7 to 10, the day 7 to 10. If you want to harmonize with Canonic, what would you choose? The 3 to 7 or the 7 to 15 to harmonize? Because also they have visit windows and they can be overlapping. But then you have to go indeed to the visit date because maybe it's not the same day. So, and also, well, for Aclara, you have the week 1, but this is the day 8. So it's quite trippy because at the end we think in similar cohorts, but they are not properly the same. Every single variable, definitely no. We have a lot of variables in the ECRF, and this is something that it happens when we design the CRF, that at the end they are noise because people, if they are not properly completed, at the end we decided to remove. And for example, here is the best example maybe. Leukocytes, we have the absolute because this is mandatory for the ED, but the leukocytes in percentage, we don't have it. And we don't have in general for all the cell population, we don't have the percentage. Because at the end we realize that people, when they enter in the data, they mix concepts, they mix percentage with absolute value, and we decided to say no. No, because at the end they will get outliers, they will get a lot of things. So we decided not to include. For example, different categories for same variables among the different cohorts. What did we do? So to maintain all the categories. Sometimes, even if we try to harmonize, it's not possible because we have the bias of the clinicians for sure, but I have emails from different clinicians saying different things and different opinions. So at the end, since no one is over no one, we decided to maintain all the categories. So a question, what if there is a mistake in the original cohort? So imagine a patient, bilirubin 14 and INR 3. Is this patient ACLF? Sure. Okay. What if the patient was taking anticoagulants or has a cardiopathy and the INR is not coagulation failure? Is this patient is ACLF? So, and what if it happens at inclusion? Because sometimes people is not, when you ask about data for the ancillaries, like, no, I need, I don't know, 100 patients ACLF at inclusion, then once that develops. So what you should do? Would you change the type of patient? Or would you consider this patient is like, okay, this is missing? So this is something that it happens with SALA. And all these patients, okay, we had queries confirming that this was a cardiopathy, blah, blah. But at the end, for them, were noise also. Because this is something that is like, is not a proper ACLF. So at the end, these patients were missing. I have to say also that in some ancillary studies, they continue including the patient, but this is something that could happen in the database. So, what if some variables or concepts have been outdated? No, to maintain the traceability, every time that a variable is considered different, we create a new variable. So, if you remember in paper 2 of PREDICT, we talk about the precipitating events, the alcohol, the infection, and so on. So at that point, we updated like the definition of alcoholic steatohepatitis, no? So this is the original one at the end. Look, at the beginning in the left side. And it was according to the subjective perspective of the physician. The patient had ASH or not. But when we applied the definition like transaminases, bilirubin higher than 3, alcohol, so some variables had outdated. So from the beginning was yes, then was no, the ASH. So at the end, also we decided it's like, well, let's to maintain, because we cannot remove what happened, because also we have published papers. We cannot remove and lose the traceability of that paper that was published at some point, but it happens. So, and finally, do you want to add combat and prospect to the integrated database, Peter Manuel? Yes. So, I have put the devil on the high angel because this is something that, well, Paolo is there also, and I did some business when we prepared the CRF. So But what is a bad condition for adding a new CRF to the integrated database? To ask recurrently the same concepts in different forms. What does it mean? For example, the readmissions, no? It's like you are asking about some complication of the patient and you say, was the patient readmitted? And this is in the events, this variable. And you said, no, but then let's put in the follow-up. Was the patient readmitted since the last follow-up? Another variable, different, no? And at the end, it's like, then data goes to Eva, for example, to Ferran, or to David, and they say, well, but how many variables of admissions we have? And I say, well, listen, 3, 4, 5, it depends. So at the end, when normally the advice is like, no, let's say hospital stays to onset date of the readmission, admission date, discharge date, why this guy was admitted, and so on. Because at the end, this is something harmonized, and it is easy to integrate and we have only 3 variables: onset, ongoing, and end, and very easy. So to avoid variables that you will miss in the future. So this is something that happens yesterday about we have to ask the score. But the point is that these variables were proposed by me last year. It's like, are you really sure that you want to avoid these variables? Because this is an inclusion criteria. Let's simplify. The physicians works very well. 1 year later, we have to modify the CRF. And it is quite tricky because we have to integrate this also. Or to think that the CRF is a medical record is the opposite. Sometimes we ask about, I don't know, I don't know how to say this, but in laboratory, we ask, let's say, all of you know that I am a smoker. I have not analyzed tobacco consumption in my 7-8 years in the foundation. But in all the CRFs, we're asking about tobacco consumption. I don't know. Let's think about the useless of several variables. And to maintain the structures of contents for each data set. It's like, that right now, more or less, we have a structure: events, vital signs, laboratories. So it's what I am saying before, is try to maintain this structure. And to ask clearly the variables that will allow to achieve your main objective. For example, combat trial, safety, no? You know that all the events should be recorded as an adverse event. And every single training I have done for combat, where the KMA is like, no, because I have completed already the event, blah, 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 and so on. It's like, it's safety. You have to complete the adverse event, because at the end, it will be the point and the goal of the trial. Well, and this one is like to follow the advice of the MC. So finally is, relax. We will find the balance between both, so we have not enemies, okay? So, and I think that's all on my side. So
you have seen that a bit of history, a bit of background, and then all the challenges of getting the data ready, and all the discussions that we can have for understanding what is the data. And now, here is a bit of a snapshot of when we try to do this type of projects, right? We call systems medicine, bring everything together. The question is, sometimes I think it's very important for us to have very clear in mind, what is the plan? What is the question? Remember writing the decision project, and we have been discussing kind of these things again and again and again. So what is the end point? It's good to, when we start this type of projects, to think about where do we want to go? And here we define some aspects, right? The stratification, we want to have some predictor for the prognosis and understanding the disease. Perfect. But what do we want to predict? And then we come to death, to transplant, ACLF, and giving you a narrative of things that we have been evolving during this project as an example. This is one example because at some point we were saying, well, we want to predict ACLF as a prognosis, or we want to predict early ACLF, which is totally different. And that was kind of an agreement that had to happen during the duration of a project. That brings back to what Christina was talking about, and that brings back also to the analysis that we are doing, because we need to modify things. And it's perfect. This is what we are aiming for, and the type of risk issues. And we want to know the response to treatment. Okay, let's make a bit of simplification, right? Let's think about that we are interested, for the sake of this presentation, on the predictors and understanding the disease. And now we have a project where we want to do multi-omic approaches for complex diseases. I think it's very important that we do one step backwards and ask this: why are we doing this, right? Because in reality, when we are thinking about the multi-omic approaches, having the different omics, what we are doing is the how. We are asking how we're going to understand the disease, using expression, using genetics, using different things. At the end of the day, we are going to try to get the what, right? The biological knowledge. But the why behind it is because we think, or we believe, that biological systems are rather complex, are highly coordinated, and having only one layer of information is not enough. We need all of them. And okay, this looks trivial. This is the why, how, what that you have seen in some TED Talks. But we try to challenge in the unit, in our unit, when we go for a project about those texts. Exactly why are we doing that? And then we move backwards. We have decided, we go for multi-omics, we have cleared the why, and we are going to do this type of profiles. Okay, but then we start to ask questions. We are talking about before the starting analysis, before the starting the project. We know the question, we're trying to ask it, what are the omics that should I select, right? And how many samples for each omic? And how many samples and how many samples for each omics? And is this, the answer to those three previous questions affected by the integrative effort that I'm going to make? And then setting the experimental design. I'm saying that because if we want to have 20 omics from blood, what are we going to do? We are going to get out 20 liters from a patient to do everything? That's not possible, right? So we need to make some kind of decisions here. And we can get some preliminary analysis. So sometimes we get lucky and there is some public data that we can do some power analysis to understand the sensitivity, etc. The point here is, with the multi-omics, every one of the omics, we have a different kind of power situation. So we have a limited number of samples, we have a limited number of budget, and we have to make decisions, right? And this is where I think gets sometimes a bit complex. Here we had to make some decisions about, what are the omics that we are targeting, and that was before where we writing the project, and that also takes time and considerations. And I'm using this symbol time and time again in the presentation because every time you see this is because sometimes we take a decision and then we need to trace back once we have new information. So we have to repeat the process of how to go for it. Now, we have done the best of what we can, right? We have selected the omics, hopefully selected the number of samples, and we go for analysis. And then maybe we analyze each one of the analyses separately. This morning I was talking on those things, or 2 hours ago. We also have to think about how we are going to do the integrated analysis, and we are going to think about interpretation. And what I'm saying is, we need to plan before we go for it, right? It's not we collect the data and then we do whatever we think is best. During the design, we have to have clear that this is going to happen. Maybe we will update the things, maybe we will do it differently, but we have to make sure that we have a plan, because that will go to the why. Now, I was saying before, right, for the independent analysis, we will do quality control, identify features of interest, function analysis per data type, they integrated? Are they omics coordinated? NWC, network analysis. And then the challenging thing, or a very, very, very high challenging thing, is going to put everything that we are getting in step 1 and 2 for interpretation, and depending on the results, how we plan to validate them. This is quite challenging. I was saying, we have a plan, right? We go for it, etc. And I'm going to use like a battle analogy, and I apologize for that, but this is, I think, no battle plan survives contact with the enemy, because this is real. As soon as we start analysis, we realize that, okay, data is not as we are expecting, this is what's happening, so we have to refine what we are doing. So now, think about this. We have defined the experimental design, the number of samples, we get the data, we have the plan, and we go for it, right? And the point is, we have the data ready. We are ready to go for it. Cristina was pointing out examples where data is ready, but we need to think about it, identify problems. We have these active communications between the data management and the harmonization, all the steps and the analysis. And sometimes we have to do analysis because there are decisions here. Every time we redo analysis, it will be 1 day, it will be 2 weeks, it will be a lot of effort. And the reasons for that could be we have outliers, missing values, low quality, unexpected. I mean, all the reasons that Cristina was pointing before. Fine. Now we have our plan, and maybe we will be kind of redoing it. And then sometimes, like in this project, right, where do we start? Every one of the groups that are doing omic analysis or generating an omic will do the analysis, and then another group does integration. We start from here, how we split the task. This is something that we had to discuss at some point during the project. And okay, let's say we take this one, each one of the omics. We conduct the analysis. We see the nature of every one of the data types. It's a very different nature, percentages, relation to a reference control. When we integrate, this is going to be different. For each one of them, we do quality control. We have a plan for the data analysis. And then sometimes, whenever we have finished it, we identify some problems or some questions on the clinical data, and we have to repeat it again because the batches were not the right ones, so we have to redo everything several iterations. I'm saying this not because I'm complaining, this is our task, this is what we like to do, right? And this is a part of the analysis. But because we want to show you that sometimes it's not just pressing a button, we have to repeat and do again and go carefully about every one of the questions. When we go to the integration, this is one of the slides that I was waiting for, because we have the overlap, then we have the different processing of each one of the data and how this is normalized. Then we go for how are they coordinated, then we do the MPC to extend, then we do the network analysis. We even write a book about network analysis. We do kind of tools or develop new tools for putting all the gene set and the function analysis together. We do the visualization. And of course, we need to repeat it a number of times because sometimes we see that the question has to be refined or things were not clear. So it's extremely fun to do this, and we do these things a number of times, and then we work with interpretation. And again, what Nuria was pointing before, and it's clear from Cristina's messages, that for those interpretations we need to have this active discussion again, all the steps what we are getting, etc. And sometimes we go back and redefine the question and go for something slightly different. I wanted to show you that in the session, we plan for those things, right? And we had this plan in the beginning, back in the Murpany 3, and we have been adapting over the time. So why we wanted to talk about those things? Because many of you are working on the lab, many of you are clinicians, you have a massive knowledge, experience day by day, a very important thing is contributing. And sometimes we don't know what you are doing, but sometimes you don't know what we are doing, right? So this is a bit of a therapy showing like what this means eventually at the end of the day. And we really want to reach the point where we all sit together with all the tools that we have, discuss the science. This is what we are doing, what we have been doing. Thank you so much. I feel like we are all learning, and I appreciate that we are all learning from you and from these interactions. So thank you to everyone, and that's to be the end.